Hey everyone, Danny here from Sidekicks. So today I'm going to begin a series of videos known as breakdowns. In these videos, I'm going to examine all of the science topics that we covered in each of our podcast episodes, as well as emphasize some topics that I believe weren't touched on enough in the videos. Beginning with our first podcast episode, getting about Guanabenz, I'm going to examine the topics of eukaryotes and protozoa, parasites, cysts, antibodies, and lastly, evolution, as well as the notion of natural selection. So looking at eukaryotes and protozoa, what is a eukaryote? So a eukaryote is a large type of microorganism that's unique in having its genetic material stored within a nucleus. This is opposite to archaea and bacteria, where the genetic material simply just floats within the cell. As well, eukaryotes are special because they have individual systems responsible for energy production. In animal cells, these would be known as mitochondria, and in plant cells, they're known as chloroplasts. The protozoa is a class of eukaryotes that's unique in being composed of a single cell. This is actually more like archaea and bacteria and opposite to the majority of eukaryotes, such as us humans, which are obviously made of more than one cell. Within the protozoa, there's a phyla or subclass of organisms that are parasitic. They're known as the apocomplexa, and an example of a member of this phyla is Toxoplasma gondii. So now going on to the topic of parasites. A parasite is any organism that inhabits a generally larger organism of a different species, known as the host, with the objective of damaging this host either directly or through starvation, so eating the host's food supply. This relationship between parasite and host is known as parasitic symbiosis, symbiosis meaning is existence together. A parasite requires a method of infection between hosts. So two examples of this would be vector infection and trophic infection. Vector infection is when the infecting parasite, known as the vector, carries a disease which it uses to damage the host. A common example of parasite vector infection would be a mosquito bite. Because if you think of it, the mosquito, the vector, transfers a disease, usually malaria, to a host with the intention of damage and usually death, for example, humans. Trophic infection is when the infecting parasite is transmitted through being eaten by a future host. So troph means to eat. Toxoplasma gondii actually transfers via trophic infection. For example, when the cat eats the docile mouse that's infected with Toxoplasma gondii. As well, in the podcast, Alex spoke on parasites such as Toxoplasma gondii being easier to evade the immune system as opposed to bacteria and archaea due to infecting extracellularly versus intracellularly. So this is due to the fact that antibodies released against parasites are only effective if they're extracellular. But what are antibodies? So antibodies are released by the immune system cells known as beta lymphocytes or B cells as part of the specific immune response. And this would all be against a pathogen, such as Toxoplasma gondii, which is just a cell that's meant to harm the host that it infects. The antibodies released against protozoan parasites, such as Toxoplasma gondii, are IgM and IgE. This would be out of a list of antibodies that are IgA, IgD, IgM, IgE, and IgG, each with a specific function. Ig just meaning immunoglobulin. The purpose of antibodies is to destroy invading cells via opsonization or the development of what's known as the MAC attack or membrane attack complex. Opsonization is when antibodies cover an invading cell in order for an immune cell known as a macrophage or a neutrophil to engulf the cell in a process known as phagocytosis. Thus, each of these immune cells are known as phagocytes. The MAC attack occurs when antibodies bind to an invading cell in order for a pore to be created within that cell, and this would then cause water to rush in due to osmosis and lyse or break up the cell from the inside out. As well, immune cells known as mast cells, basophils, and eosinophils are also act as defense mechanisms against parasites. Eosinophils are especially important because they release what's known as cytotoxic granules against parasites, which cause them to commit apoptosis or programmed cell suicide. In addition to this, Toxoplasma gondii can be specifically destroyed by immune cells known as macrophages via phagocytosis. Moving on to the topic of cysts. So cysts, for example, the brachiocytes and tachyocytes within Toxoplasma gondii, are simply just groups of cells enclosed within a membrane. In the case of Toxoplasma gondii, however, replication within felines occurs when the cells are only as tachyocytes. And the majority of brachiocytes of Toxoplasma gondii are found within the central nervous system, which is composed of the brain and spinal cord, and it's the largest part of the nervous system responsible for basic organism function. The conversion between tachyocytes and brachiocytes in Toxoplasma gondii is due to stress and occurs via the phosphorylation of what's known as the EIF2 alpha eukaryotic initiation factor. So the phosphorylation of EIF2 alpha causes what's known as the integrated stress response, or ISR, within an organism. Lastly, looking at the topic of natural selection and evolution. So in the podcast, we spoke of the ability of Toxoplasma gondii to render mouse hosts immobile in order for them to be easily eaten by cats, thus transferring Toxoplasma gondii from the infected docile mouse to the cat as being an evolutionary trait or mutation. In essence, this is really an example of Charles Darwin's famous notion of natural selection, the ability of a member of the species to survive and eventually reproduce based on changes in its phenotype or traits. So these changes in phenotype occur through the adaptation of the member of the species to its environment. Looking at this through the lens of the podcast, in order for Toxoplasma gondii to survive and eventually reproduce, it must be able to transfer from host to host. Since when within rodents, Toxoplasma gondii transfers to cats via consumption of the rodent by the cat, it manipulates the minds of rodents that it infects to make it yield to the predatory nature of the cat. This then ensures that Toxoplasma gondii will continuously survive and reproduce so long as it infects rodents. Well, that's all for today's breakdown video. I hope you guys enjoyed, and we'll tune in next week where I release a breakdown video on the second podcast episode, Tackling TB. As well, before I go, I would like to remind our viewers that in every podcast episode, under the description, you can find really cool facts and trivia, and I encourage you all to please read those to be interested in the many facets of all of these topics that we speak of. Thanks everyone, and I'll see you later. Danny out.